Hey everyone, and welcome to this explainer. Okay, think about your closet at home for a second. If it's anything like mine, it gets messy fast, and organizing it requires some kind of system, right? Maybe you sort your stuff by color or, by season. Well, I want you to imagine that exact same closet, but instead of clothes, it's packed with millions upon millions of vastly different, completely wild organisms. That is exactly what biological classification is. It's the ultimate organizational challenge, sorting the impossibly huge, messy, beautiful closet of life on Earth.
Seriously, think about it for a minute. If you were tasked with organizing every living thing on the planet, where would you even begin? Do you group giant blue whales with tiny goldfish just because they both swim? Or do you stick a mushroom in with an oak tree because they both happen to grow out of the ground? Early scientists had to grapple with this sheer, mind-boggling scale and diversity of life. They didn't have our modern DNA sequencing tools, so they just had to start somewhere.
Okay, let's dive into this. Here is our roadmap for today's journey. First, the quest to classify life. Then, flaws in early systems. After that, the five-kingdom system, followed by Monera and Protista. Then we'll look at fungi, unique decomposers, and finally, plants, animals, and beyond.
Part 1. The Quest to Classify Life. From Instincts to Aristotle.
So, before science as we know it even existed, humanity's earliest classification systems were purely instinctual. It was all based entirely on survival, what could be used for food, what worked for shelter or clothing. But then, ancient Greece comes along, and Aristotle makes the earliest attempt at a more scientific approach. He used simple, visible traits. He separated plants into trees, shrubs, and herbs. And for animals, well, he had a very relatable, albeit kind of flawed, system. He literally just divided them into those that had red blood and those that didn't. Eventually, this basic idea evolved into the 1700s when Linnaeus developed the famous two-kingdom system, Plantae and Animalia.
Part 2. Flaws in early systems. The categorization crisis.
For a long time, Linnaeus' two-kingdom system of just plants and animals worked well enough. I mean, it was super easy to understand. But as technology advanced, a massive categorization crisis started brewing. On one hand, you had this simple old view of plants and animals, but on the other, a growing mountain of confusing outliers. With the invention of microscopes, scientists were suddenly staring at entirely new worlds of bacteria and single-celled organisms. What on earth do we do with microscopic life that completely ignores the plant and animal boxes? A vast number of organisms just didn't fit. The two-kingdom system was breaking down.
And this breakdown led to some truly bizarre biological roommates. Because early classification relied heavily on just the presence of a cell wall, organisms that had absolutely nothing else in common were lumped together. The old system awkwardly grouped simple prokaryotic bacteria right alongside complex eukaryotes. It lumped single-celled organisms with multicellular ones. And perhaps most awkwardly, it forced non-photosynthetic fungi to room with towering green autotrophic plants. Imagine grouping a delicate little mushroom with a giant redwood tree just because they both happen to have a wall around their cells. It was a total mess.
Part 3. The five-kingdom system. Whittaker's hero moment.
So, a new framework was desperately needed to sort out these mismatched roommates. Enter R.H. Whittaker. In 1969, he stepped in to solve this categorization nightmare. And he didn't just look at one single trait like a cell wall. He established a comprehensive set of highly scientific criteria. He looked at cell structure. Is it complex or primitive? He looked at body organization. Is it single-celled or multicellular? He examined their mode of nutrition, how they reproduced, and, absolutely crucially, their phylogenetic or evolutionary relationships.
And this brilliantly illustrates the solution. By applying these strict criteria, Whittaker gave us the five-kingdom system: Monera, Protista, Fungi, Plantae, and Animalia. You can think of this breakdown as the ultimate biological cheat sheet. It so cleanly separates the diversity of life. Monera captures all the simple prokaryotic cells without a nuclear membrane. Fungi finally get evicted from the plant kingdom because, while they do have cell walls, theirs are made of chitin, whereas plants use cellulose. It really represented a massive leap forward in human understanding.
Part 4. Monera and Protista. Into the microscopic world.
Okay, let's zoom all the way down into the fascinating microscopic world. We'll start with Kingdom Monera, which consists exclusively of bacteria. They are far and away the most abundant microorganisms on Earth, and while their cellular structure is structurally simple, their behavior is wonderfully diverse. They generally come in four distinct shapes: the spherical cocci, the rod-shaped bacilli, the spiral spirilla, and the comma-shaped vibrio. But hey, don't let those simple shapes fool you. Their metabolic diversity is absolutely unmatched. Within Monera, we have characters like the archaeobacteria. You can think of these guys as the rugged, extreme survivalists of the microworld. Because they possess a completely different cell wall structure than other bacteria, they thrive in the most hostile environments imaginable. We're talking boiling hot springs, ultra-salty areas, and marshlands. They even live in the guts of cows and buffaloes, where they're responsible for producing methane gas.
Now, contrast those extremists with eubacteria. These are known as the true bacteria, like the cyanobacteria nostoc, for example. These true bacteria play massive, absolutely essential roles on our planet. They recycle vital nutrients like nitrogen, iron, and sulfur. Nostoc can literally pull nitrogen right out of the atmosphere using specialized cells. Without them, the nutrient cycles of Earth would just grind to a halt.
Stepping slightly up the evolutionary ladder, we enter kingdom Protista. The crucial takeaway here is that Protista acts as the vital evolutionary link. They are still mostly single-celled and primarily aquatic, but unlike bacteria, they are eukaryotes. This means they possess well-defined complex nuclei and membrane-bound organelles. It's a really broad kingdom that sweeps in chrysophytes, dinoflagellates, euglenoids, slime molds, and protozoans.
The visual variety in this group is just incredible. On one hand, you have ocean-dwelling dinoflagellates. They wear these stiff cellulose plates like literal armor and can multiply so rapidly that they turn the sea red, releasing toxins in what we call a red tide. But then, on the other hand, you have protozoans like the paramecium, which acts like a tiny predator, actively moving through the water using thousands of tiny, coordinated hairs called cilia to steer food straight into its gullet. The diversity is truly stunning.
Part 5. Fungi, unique decomposers.
For centuries, fungi were the misunderstood characters of our biological story, awkwardly shoved into the plant category. But Whitaker finally gave them their own kingdom, and here is exactly why fungi are definitely not plants. First, they are entirely heterotrophic, meaning they cannot make their own food. They absorb soluble organic matter from dead substrates, acting as nature's ultimate recyclers. Second, and this is a fantastic fact to remember, their cell walls are made of chitin, not cellulose. Chitin is the exact same tough material that makes up the exoskeletons of insects. No way, right? And finally, their bodies aren't made of traditional tissues, but rather a network of long, slender threads called a mycelium.
This kingdom covers a huge spectrum that actually connects directly to your everyday life. It includes mucor, which is that fuzzy bread mold you might find if you leave a sandwich out for way too long, but it also includes agaricus, the delicious mushrooms you might order on a pizza. They are cosmopolitan, occurring in air, water, soil, and even on plants and animals, just silently recycling the world around us.
Part 6. Plants, Animals, and Beyond.
So by finally sorting out all those microscopic and fungal outliers, science could properly define the macroscopic kingdoms we're most familiar with. Kingdom Plantae contains all the eukaryotic, chlorophyll-containing organisms. They have cell walls made of cellulose and are almost entirely autotrophic, making their own food via those prominent chloroplasts. Kingdom Animalia, on the other hand, contains organisms that lack cell walls entirely. We are heterotrophic, relying on others for food, and we possess highly advanced tissue and organ systems. It's a clean, logical separation that finally makes sense of Linnaeus's original two groups.
But you know, here is the most exciting part of science. It is never truly finished. The five-kingdom system is brilliant for sure, but it actually completely excludes non-cellular entities. We're talking about viruses, viroids, and prions, as well as symbiotic organisms like lichens, mostly because they simply don't fit nicely into conventional cell theory. So the story isn't over. As you read the source text for this explainer, you'll note that an even newer system is already emerging, a three-domain system that actually splits the Monera kingdom of bacteria into two entirely separate domains, giving us a brand new six-kingdom paradigm.
Ultimately, our categorization is only ever as good as the technology we use to observe the world. From Aristotle's simple eyes to early microscopes, all the way to modern DNA sequencing, our view of the biological closet is constantly changing. So I'll leave you with this. As our genetic and evolutionary understanding deepens, how will we redraw the map of life next? Thank you so much for exploring the incredible complexity of life with me. Keep wondering and keep learning.